Hello and welcome to Unsupervised Thinking, a podcast on neuroscience, artificial intelligence, and science more broadly. We are a group of computational neuroscientists. I'm Grace.
I'm Josh.
And I'm Jan.
And the topic for this episode is brain organoids, which are basically little blobs of brain cells grown in a dish. And we'll get into what they're used to do and how they're grown and all of that. But first I want to say that this topic was recommended as a topic for us to do by Marius Martin. And he even recommended some readings, which I think is great. But I think he was a little optimistic about our reading comprehension for basic biology. So I found a few gentler reviews to get us started. And so the first of those is Brain Organoids, Expanding Our Understanding of Human Development and Disease. And that was published in September of 2018. The first author is L.B. Chua. And then the second one is Cortical Organoids, Why All This Hype, with the first author, Fabio Marciner. I find it hard to read that title in like a neutral tone.
Yes, but surprisingly or slightly confusingly, that article was actually very positive about organoids.
Oh yeah, no, they told you why all the hype.
Yes.
Yeah, yeah, so those are the two review articles we read, and then we also read this interview with a bioethicist named Sarah Chan, and that was on eurostemcell.org. And we looked at this very recent New York Times piece by Carl Zimmer that was called Organoids Are Not Brains, How Are They Making Brainwaves? And so that gave an overview of maybe some more recent research as well. Because I guess as we'll get into, this is a pretty recent thing for a lot of people to be exploring. So maybe we can start by just talking about like where this falls in the space of neuroscience research. So I guess the vast majority of neuroscience research is done like in a whole animal. You grow a full animal and you look at its brain either while it's still part of the animal and the animal is doing a task, or potentially you grow a full animal and then you take the brain out of it and you dissect it, stain it for different cell types, that kind of thing. So that's, I would say that must be the dominant research amount of research in neuroscience.
Yeah, I mean, I think that is our like kind of bias in terms of where we're situated in the field. But I think human neuroscience is like
Humans are animals.
Yeah. Well, yeah. I mean, mostly though it isn't non-human animals. It's mostly like I think by number, and like neuroscience is predominantly studies of humans, and I think things that are generally clinically relevant. I mean, then there's sort of like basic animal neuroscience is I think some sort of subset of neuroscience, but certainly in terms of people who are looking to do more basic research, it's primarily done in animals. And then I guess the dominant animal is presumably rodents, and again, mostly in the context of clinical research. And then some subset that's significant because it can be used for clinical research is like cellular and molecular research on neurons. So this would be more about like what genes do neurons express, how do development pathways of specific of single cells or small numbers of cells, how are they affected by genes, and how are they affected by disease processes, and that's kind of where we're situating ourselves with this kind of research. So this is research that's less about what do brains do and more about the sort of cellular and molecular properties, what proteins are expressed, what genes are responsible for the proteins expressed in cells, and how they can go wrong in diseases, I guess is kind of the broad situation we're situated in. And then I guess with organoids, it's a more recent topic where you can, I mean, so for a while people have been able to culture neurons in a dish. And with organoids, you're able to go, instead of just taking a bundle of a small number of neurons and putting them in a dish and running potentially kind of, molecular experiments on the neurons in a dish, again, not really functionally recording from them or seeing what they would do in an animal setting, more just like how do chemicals affect their growth. Now you can do this with clusters of neurons that are both larger and potentially more like, in certain ways that we'll talk about, neurons that you would find in animals.
And yeah, so normally when people do culture experiments in neuroscience, like they're just working with neurons in a dish, it's my understanding they still start from neurons in a full animal, take them out of the animal, and then grow them like grow more of them on the dish, right? So you're still starting with the full animal. You're just deciding to take a small part out of it and study it in a dish. Whereas with the organoids, you're not starting with the full animal. That's one relevant difference. And so the other thing, and this was the thing that I thought was the main difference and why I didn't understand why all the hype when I was first hearing about this. One of the things that gets highlighted a lot is that they're three-dimensional. And normally when you grow cells in a dish, you grow them like on a flat plate and they just grow like next to each other, but they don't form structures on top of each other or in any interesting way. So the other aspect of this that's different is the structures that can form from these organoids.
And I mean, I think cell cultures also, from my understanding them as well, they kind of, you have to like, they have a very limited lifetime. I'm not sure if this is the case with organoids as well. Like, because you're taking them out from, you're starting off with like these cells that are in an animal, and you're taking it out of like an environment that it's very comfortable in. You've sort of perturbed it massively when you're doing that, and you're basically just feeding it with all this stuff that you try to keep it alive as long as you can to do these experiments. Whereas I suppose with organoids, since these cells have developed from stem cells or the like, then I imagine that they're much more well adapted to the environment that they're in when you're doing these experiments or whatever, compared to cell cultures. But I guess this wasn't actually really talked about, but I would imagine that's the case.
It is the case. The longest experiment with these organoids that I noticed in what we read was over 1.5 years.
Yeah,
so that has to be longer than
definitely.
normal
Like cell cultures, I think from some colleagues that did it, I think it's like 2-3 weeks, I think is
that's pretty different.
Yeah.
So we can get into how they're grown. So in this first review that we read, though, they kind of had this backstory about neurogenesis in the brain. So the traditional story for a long time was that the adult brain has no new neurons grow in it. You're kind of born with all the neurons that you're going to get. And then more recently it was discovered that there is adult neurogenesis. Certain parts of the brain can produce new neurons. There was this interesting bit that I didn't know, which is about how this was proven or studied in humans in particular. And that was that they basically used this, I guess it's like a tag that would tag a cell if it was dividing. So it was like becoming a new cell. And they put it into the brains of terminally ill cancer patients because they needed to put it in, like normally in an animal experiment, you kind of put this tag in and then you'd sacrifice the animal and you'd check where it showed up in the brain. And I think the part of the important bit here is there's kind of a time window where you need to, between when you put it in to when you look. And so basically there was they had to find people who were like, well, you're going to die soon, so we can put it in you, and then when you die, we can look at your brain. So these people were like, yeah, we'll participate in this experiment. We know we're going to die. You can inject us with this and look at our brains afterwards. And so that was one of the ways that they found out that adult neurogenesis does happen in humans.
And there's this nice quote from, I guess, Santiago Ramon y Cajal, which is a very, very kind of old neuroscientist. Apparently he had a dogma saying everything may die, nothing may be regenerated. So this neurogenesis discovery sort of proved that wrong.
Yeah. But as I understand, after reading the rest of the review, the fact that adult neurogenesis happens has no bearing on the building of cortical organoids or the fact that we can make these cortical organoids. Is that correct?
I mean, maybe it makes sort of the brain a more amenable organ to grow compared to other organs that don't have genesis.
I mean, as we're getting into where this comes from, I mean, ultimately they're going to use stem cells, right? This is what we're sort of prefiguring. But I just have not been, sort of abreast of what's been going on in organoids generally. And they kept on using cortical organoids, and so I was looking, and it's clear there are other organs that you can make little, kind of versions of in a dish now.
I think those were a lot earlier than brain.
They were earlier, but they weren't so much earlier to some extent because they had to start. I mean, so a lot. Okay, so we kind of have to get to the backstory to get into this. I the way I sort of personally remember this happening is people got very excited by the use of stem cells in like the early 2000s.
Yeah, and there's like embryonic stem cells that actually come from an embryo, and these are undifferentiated cells that, you know, are the cells that turn into all the different types of tissue in the body. So they have a lot of power in them. If you have them and you put them in the right environment, they'll turn into the type of tissue that you want.
Mm-hmm.
And during the Bush era, there was ended up being in the U.S. a moratorium on the use of embryonically derived stem cells.
Because it does assume that you had an embryo that you're using for this purpose and not letting grow into a human. It was a moratorium on human.
Yeah,
stem cells.
human embryonic stem cells. I remember this being like an ethics debate, and we'll get into the ethics of it later, although the ethics kind of haven't changed. It seems like the people who would now have any ethical qualms with organoids maybe would be the same people who had them then, just under a slightly different guise.
Well, but I thought that there was a development maybe 3 or 4 years ago, or maybe longer, where you could get stem cells not from...
Yeah, so the point was, motivated by this moratorium in the US, a bunch of researchers figured out how to induce stem cells from somatic cells. So people were able to figure out how to take cells from humans that are like from not embryos and turn them into pluripotent stem cells. So this is induced pluripotent stem cells.
Yeah, so one of the examples they give I think in the New York Times piece is they take it from the skin. Mm. So you can take like a skin cell and basically you like reverse the clock on it and take it back to a time when it was a pluripotent stem cell, and then you put it into some environment that can turn it into something else and it can proliferate from there.
And to kind of get like the sort of very high level version of the context again, people who culture these stem cells in dishes have figured out a bunch of like recipes or sequences of chemicals to expose them to that allow them to do various things. See how these cells develop, see how they, are subject to diseases. They can infect these cells with viruses. So this is where we're headed. And yes, increasingly they've been able to sort of turn them into cells that look like various specific body type cells. And that includes other organs. So you can do this for the liver. They can do this for the stomach, like the cells that would line the interior of the stomach, or like line the interior of the esophagus, or other organs. And cortical organoids are in some sense only like one potentially interesting for neuroscientists, version, but one category of use or specialization of cells that you can induce from these pluripotent induced pluripotent stem cells.
But there's also, so one step is getting from the pluripotent stem cells to like the specific cell type that you want in that organ. The bigger challenge seemed to be for a long time getting it to, as Grace kind of pointed out earlier, getting it to like actually have the full structure of an organ as opposed to just a tissue sample from a specific organ type.
Yeah, these are like all obviously contributions, but sort of the sequence of contributions, I guess, was that figure out some chemical recipes to make it approximately start turning into a certain cell type, and then apparently big innovation that like really made automatic differentiation of cells into like a real meaningful structure happen was like putting them in 3D. So in an animal when it's developing, you don't just have like cells flat on a plate. And basically in like the real brain or in the real body, generally there are like extracellular matrices and things like this. So the sort of, I think, way that this is thought of is that if you build a sort of scaffolding out of some chemical structure that you put the cells in, they can kind of grow on it as though it's like an artificial extracellular matrix kind of scaffolding. So like my visualization of this mentally is that there's like a 3D grid of sorts that you build out of some polymer or something like this, natural polymer. And
so the extracellular matrix is a thing that exists in the body. And so the story that they tell as the backstory of, there's actually like a few different times where organoids were invented, it seems, or kind of methods for creating them were rediscovered. And so one of them is that in 1990, someone named Mina Bissell was trying to grow breast tissue and couldn't get a growth of the tissue that would actually produce milk. So you can't study kind of the function of it in the dish if you can't get it to do its main function. And then they realized that if they add this extracellular matrix extract that presumably is, I guess, from the body, like they take it from an animal and put it onto the dish, then that provides this scaffolding that is normally there for the cells. It provides that in the dish, and then they can kind of grow into the more proper shape. And I was just kind of surprised that, like, well, I guess maybe, so this was in 1990, so this wasn't using stem cells, I assume, because...
Or it wasn't using an induced stem cell.
Yeah.
I don't know, maybe was it actual breast tissue that had been extracted, or was it like a embryonic stem cell that they had turned into breast tissue? Unclear.
No, it was transferred from a mouse. So it was cultured just like kind of normal culture. You take the tissue from the animal and you put it in a dish, but then it wasn't working right in the dish, and so they put in this extracellular matrix and got it to actually produce milk. And I guess, I was just kind of surprised. I don't know how long they've been doing this for, but it just seems like, you put in the extracellular matrix. Like, if you care about structure, I guess it seems kind of a natural thing to try, but it seems like this happened, and then people didn't do that for a while until like 2004 or something was the next time they really like tried to make this kind of three-dimensional structure happen in these cultured tissues.
I don't really so what creates the extracellular matrix in animals?
Yeah, so I think it's like, it would be, this is where it would actually be useful to have someone who has a sort of more visceral intuition for what this is. But my sense is that it's actually, you don't have to think about it as like being a very precise structure. It's more like a gel or something. Like the way when you produce agar, like for the base, for the bottom of a plate. So like agar is like a basically a starchy like substance that you can make like a gelatin out of for the bottom of plates on which you would culture bacteria or cells. And you can do something broadly similar where you can make kind of a substance that's kind of like a gelatin-like substance that you put the cells in, and that becomes the cellular the sort of the extracellular scaffolding. In the context of these cultures, it's still like a medium in which the cells are placed rather than something that's like highly precise.
Yeah.
According to Wikipedia, it is a three-dimensional network of extracellular macromolecules such as collagen, enzymes, and glycoproteins. So yeah, I feel like it's kind of like a bunch of stuff that comes out of cells and mixes around and everything.
I think it is kind of surprising that just like you just throw in that and then suddenly it kind of works. I mean, especially when, I think so we'll talk about how well it works with brains and stuff, but with things like very structured organs like livers or kidneys, are relatively structured. They have like properties which are very similar to like the biological properties of livers. I think they made a gut recently that could actually host like bacteria, the same bacteria that you see in guts, just by having the right scaffolding, I think, as far as
I saw this as well.
So like I found that like pretty surprising as to how easy it was once you added that step.
Yeah, something that they say in this story about the milk production is, and they just kind of like mention it casually, but it seems relevant. They're claiming that the environment activated like a genomic program. Like the fact that they were in the extracellular matrix, it wasn't just that it physically provided a place for the cells to grow. It was like being in that environment led the cells to activate certain genes and whatnot to change their activity. So it's not just about the physical thing that it's in. It's about how that interacts with the cell's own processes. I'm a little... the history's a little complicated because all these things kind of got like redeveloped and stuff. But I'm looking at a bit that says that at least in 2014 and 2012... they were using this kind of thing to grow neural tissue, essentially.
Also, the guy that did it first in intestinal, liver, stomach, lungs, and prostate organoids is named Hans Clevers. I know, I love that name.
Yeah. But yeah, so they're saying that these early cultures for neural development were called neurospheres. And yeah, so you just kind of like put the induced stem cells into the right kind of mixture, and then they would turn into not just neurons, but astrocytes and oligodendrocytes, which we talk about in our glia episode, which are kind of like quote-unquote supportive cells. So they're really like kind of, it's not just that you're growing a plate of neurons, you're kind of growing most of the types of cells that you'd expect to exist in neural tissue.
Yeah, and so I think this is what's considered the big deal, both when you're generating other organoids and cortical organoids, is that by putting these pluripotent cells in this, kind of appropriate situation where they can build the 3D structure on a kind of molecular scaffolding, you basically get differentiation into a bunch of different cell types that represent what you would see in the real organ. And not only do you get the cell types, but perhaps surprisingly, you actually get some of the macrostructure of how the cell types lay relative to one another. So in the case of the surface of the stomach, you get some of the sort of folding that you would get in the surface of a real stomach. And in the case of the cortex, you get a kind of stratification of cells into essentially layers by a certain kind of type that resembles the kind of layering you see in the real cortex of an animal.
If you look at the slides or the images, I guess, in the review, I think if you had a glance at that, you would think that's like a slice of brain tissue or something.
At least certainly a casual look at it.
Yeah.
It seems very plausibly like what you would get in a real brain slice.
And it's also interesting that the way you kind of mentioned it with the sort of the folds of the stomach, you can also, there was one experiment they did where they applied pressure, just mechanical pressure to certain parts of I think the top layer of one of their brain organoids, and it started exhibiting this sort of folding as well that you see in cortex, which is quite cool.
Yeah, they were talking about this gyrification, the wrinkles that exist in the primate brain. And yeah, if you again, it's a lot of it is if you put it in the right environment, either chemically or in this case maybe more mechanically, it seemed like they're applying pressure and stuff to it. You can actually get a lot of the right things to happen, which was pretty surprising to me. The fact they talk about, you get different cell types, you get a bit of the layering in cortex, so cell types kind of clumping in different layers that you can see by looking at the slice through a microscope. You get things that relate to hindbrain and forebrain and kind of these precursors of these kind of evolutionary distinct brain areas that are very well defined and known in neurodevelopment. Even in the early examples, they talked about, so they were first doing it and they could get the cells to differentiate into different cell types, but it wasn't showing much structure. But then they found a way to get a neural tube structure to develop, although the cells weren't differentiating that much. But yeah, just kind of, you put it there and then the normal developmental process happens?
Yeah, so, I mean, getting to this explicitly, I think part of, I think from a certain perspective, this could be seen as surprising. It's you just put some neurons in the right kind of medium and they grow. But I think the perspective that I take that somehow makes this a little bit less surprising is that's how animals grow really anyway, right? You take a few cells and you put them in the right kind of medium, like in a uterus of a mammal or in an egg, and they differentiate and develop into a whole brain. So we know that a small number of neural cells can turn into a brain because that's how it happens. Neural progenitor cells can turn into a whole brain because that's how it happens in real animals. So the fact that, I mean, again, it's slightly more removed. There's not the other kinds of cells of the body, but at the same time, these don't turn into real brains. They turn into these kind of weird partial brains that have only some of the structure. So I mean, I do feel like one has to consider this relative to the normal developmental process that happens when cells from an embryo turn into neural progenitor cells and then differentiate and coalesce into a brain of an animal as it's developing under normal circumstances.
I mean, it doesn't make it less cool, first of all.
Yeah.
Or sci-fi, kind of, And also, I mean, you're removing everything else. I think you are starting with a lot less than what you get when you're not getting all the nutrition that you get from.
So they are providing nutrients.
Well, yeah, but you don't know.
I mean, even then, I was surprised when I first heard about cell cultures and keeping them alive just by giving it some mix of nutritious liquid. I mean, that's kind of weird as well. And I think this sort of extends the sort of
the weirdness,
yeah.
Yeah, no, I was surprised. I mean, yeah, so you have to provide them in this witch's brew of things you have to give them. You have to give them things that signal to them what to turn into. And you also have to provide nutrients, and there isn't a vasculature system in
exactly,
organoids, so you have to be providing everything that blood would normally be providing. And they also talk about how they're grown in like a low gravity situation by like spinning them or shaking them a lot of times, which I guess the womb is kind of low gravity from the perspective of like a developing fetus and stuff. But no, I'm still surprised. Like, just it still seems like it happened pretty quick to figure out how to get this far, especially when you think about just how decontextualized it is and how little we know about the molecular details of almost anything. That we could kind of get it right well enough for as far as it goes, for as far as it does go in terms of turning into different cell types and different very early developmental structures and all of that. I'm still surprised. Again, I was starting from the belief that organoids were simply three-dimensional neural cultures, and so I had very low expectations. I thought you just grew normal neural tissue in 3 dimensions, and people thought that it was going to be special.
Yeah, I mean, I'm maybe coming from the opposite, kind of where's my jetpack kind of expectations that people would have had in the past when thinking about the future, which is like, can we grow an animal in a test tube, like a whole animal? I mean, the answer presently, as far as I understand, is no. But if you could expect that you could take an embryo and put it somewhere in like an artificial incubator and have it grow into an animal, I don't know how different that is than expecting that you could take part of an embryo and put it in an artificial incubator and have it kind of grow into a extremely broken version of an organ.
The part of the embryo, though, is part of the reason that it's even more surprising to me. Like, if you took a whole embryo and just put it in a container with the right nutrients, maybe I'd expect it to work even better. But the idea that you can just kind of pull a little tiny bit, and apparently the development of the brain doesn't need so much interaction with all the other parts of the embryo as it's developing.
Well, but it clearly does need, like...
yeah, they're not perfect. Well, okay, they're very far from perfect.
But yeah, so they're so far from perfect. Like, I mean, again, this is just about expectations.
Yeah.
Right? It's like a real embryo... at some point the brain starts developing, and I mean, I am not a developmental biologist, but it's interacting with other things to some extent. Presumably it's interacting in terms of getting nutrients from other parts of, I mean, it's, sort of passed to it or diffused to it, however this is happening. And it's also the case, I mean, certainly as it develops, it's actively getting nutrients transported to it, which here are sort of done by proxy of a fluid-filled bath or whatever that's being circulated artificially. But they also are getting mechanical constraints imposed by the growing body. And here the mechanical constraints are considerably more variable, and as a consequence the morphology of the nervous system that's growing in a dish in these artificial settings is more variable as well. I do feel like we have to remember that this is something that grows normally. So it isn't, and it isn't the case. And actually, I mean, in listening to some talks on this on the internet, it becomes clear that it isn't the case that it's like scientists making these things grow. I mean, yes, it's the case that you provide them with the right nutrients, but in some sense, these things grow on their own. And if we should be surprised, it's that these things grow on their own as well as they do outside of their normal circumstance. But they are, in some sense, recapitulating a mostly normal circumstance, like partially. Sure. In a dish, right? Yeah.
Sure, that's fair. That this is what these cells are supposed to do.
Yeah. But I mean, if you look at the mechanical support they're giving it, it's very rudimentary so far. It's presumably very early days, and like, I guess they're probably experimenting with 3D printing all this fancy stuff, which, is more faithful to the structure of the brain maybe, and seeing how that works or something, I don't know.
Oh, yeah, and also it was the case that, as I said, the longest study went out to like a year and a half, and even shorter than that. So they were looking at, I think, gene expression over time in these things. And it was the case that gene expression, like about a month in, matched the first trimester of gene expression in a actually developing fetus in a womb. And then like 2 to 3 months in or 3 months in, it matched the second trimester. And so, like, they're developing even on the proper time scale, it seems, which was surprising. And this came up also with the more recent work that was discussed in the New York Times article, that if you monitor the electrical activity, it shows oscillation patterns that also follow a roughly similar time scale as actual fetal development.
Yeah, so I guess, now, so we've kind of debated the question of, is this surprising or cool? Now, with that, we can ask, is this useful for neuroscience? Because what you just mentioned about, like, the fact that it matches the gene profiles that you might expect in development, and that it maybe exhibits some sort of functional electrical activity, that kind of is also quite an interesting question to ask, I think, right?
Yeah, and the first review that we read does focus a lot on the use of it for the study of diseases. And so I guess, the main areas, or really it's one area, this is best used to study development. That's what you're looking at, the very early stages of brain development. And then
so when you say diseases, you mostly mean sort of diseases where the signatures of them are visible at the level of single cells or over developmental trajectories.
That's what I believe, but the review we read talks about diseases where that's not true.
One canonical example that came up a few times in the readings was Zika, right? So Zika, as sort of is fairly well known at this point, it causes a kind of microcephaly, so like a shrunken brain, especially for basically when babies or pregnant women get this.
It's a mosquito-borne disease, and if you're pregnant, when you get bitten by the mosquito, your baby might be born with these, like, the stereotype is this very small head because the brain is shrunken.
And so here, and this is sort of a very striking example of a success of cortical organoids, at least as a model of a simple model of a neurodegenerative disease, or rather not a degenerative disease, but a developmental disease, is that you could grow an organoid and infect it with Zika, and it will not grow as well. It will produce like fewer neurons and produce a smaller clump of organoid brain. So you basically can reproduce as a model of this developmental disorder. You can use organoids to sort of capture the most salient property of that, which is not as many neurons grow, and the brain volume size is thereby reduced.
Yeah, and I want to highlight again that this is using human neural cells, which is very relevant when it comes to this kind of disease treatment, and especially like this, the ideal with the Zika is some kind of rapid response, because it started as like an outbreak, and you want to be able to test out different treatments for it and understand how it progresses kind of as quickly as possible using the actual types of cells that it's affecting, which is human brain cells. So that's a benefit of the organoids compared to animal research. And they actually go into a few detailed ways in which, like, the typical, mouse model of studying disease or studying drug treatment just, like, cannot work. So in the case of lissencephaly, which is not a shrunken brain, but a smooth brain, because as we said, like, the primate brain is normally wrinkled. This was another thing that was studied in these organoids, and you can't study that in mice because their brains are naturally smooth. That's just, like, they don't have the wrinkles that we have, so you can't use them as a mouse model of that. They actually they refer to mice as both lissencephalic and microcephalic. Like, they have small brains, but, like, I feel like they're the right size for their body. Like, they're small compared to us. So I thought you could still study microencephaly in mouse, but I guess maybe not. But yeah, so that's another thing that's kind of, you want the human cell. They also actually just had this whole list of like drugs that have different effects in mouse versus human. Like, they said that aspirin causes birth defects in mice and rats, but not in humans. And they also talk about aspartame, which is like fake sugar, and that causes lymphomas in rats, but doesn't seem to cause it in humans. So sometimes when you see like a story in the news, like, fake sugar is going to kill you, it may have been done in an animal where the findings actually aren't transferable. So yeah, there was a lot of interesting things that they highlight about not just like the diseases that you can't study in mouse or other animal models, but also the fact that the drug effects won't be the same. And so you really need to be using human tissue to get at treatments for human diseases, and this is a way to do that. And then there's also the fact that because you can take, a skin cell of a person, turn it into a stem cell, and grow the cerebral organoid, that opens the door to personalized treatment. So you can, they talk about doing diagnoses in this, like if you grow some organoid tissue for someone based on their own stem cells, you might learn something about the disease that they have, and then also testing drugs on that tissue specifically to see if that person will respond well to it. And I think even like potentially growing tissue to put back into their body. So that's like crazy, like personalized medicine kind of stuff.
In neuroscience, I think one of the issues that is talked about a lot is just the failure of clinical neuroscience to actually come up with cures or even explanations for common sort of disorders like schizophrenia or things which are, I think a big reason is that they're systems level problems basically, right? And it's hard to look at the system of a human because you can't really invent it that well, and you can't really look at systems level stuff when you're doing cell cultures because there's just not enough structure there. So I think part of organoids, which is quite exciting, is in trying to get a bit further along the lines of models of systems level disorders. There is a little bit of an early study here, which essentially they look at models of schizophrenia, and they make organoids, and they basically, it's in a very rough sense systems level in that they're looking at how the cells interact in some way, and they find that basically there's two different cell types that usually in control subjects, one cell type migrates to one sort of brain area during development, and then the other cell type either is more dense or less dense in another area. And then for the schizophrenia model, there is essentially a disorganized migration of one cell type compared to the other, and this sort of leads to what they claim is the problem with schizophrenia. It's a very early kind of proof of concept, but I think it's quite interesting.
Yeah, I went through a range of emotions with this because so they talk about schizophrenia and they talk about autism as well. And they kind of do this thing that happens in clinical neuroscience a lot where they throw out these cellular level problems to explain these very complex diseases that, we don't even know if everyone diagnosed with the same thing really has the same underlying problems and all of that. And so they were talking about like, autism is potentially an imbalance between excitation and inhibition in the brain. And it's like, well, so is like epilepsy. So it's tough because it doesn't feel like necessarily what you can learn from looking at this low cell level behavior, like how exactly it's going to relate to the disease. But then at the same time, again, so in some of these cases, they're taking stem cells or they're taking cells that they turn into stem cells from people with schizophrenia, from people with autism, and just it's almost even like not even trying to make so many claims about what the cause of the symptoms are. It's just, what do these brains look like? Like, how can we get at the development that happened in people with these genes? And then you can say, okay, well, this is what was different about the people who had autism, about how their organoids developed. And maybe you can say that that's why they have whatever symptoms they have. But even if you can't directly relate it because you're not reading out behavior in these organoids, they don't actually do anything, they're not in a full animal. Just even knowing how genetic differences lead to brain differences is something for sure.
Yeah. I think it's especially important when you consider that most of these disorders have tons of genetic sort of correlates, and they're all kind of interwoven in some weird way. And so there's this, I just came across the phrase in the review there, like this watershed hypothesis when it comes to, is it autism spectrum disorder?
I think they said it for autism and schizophrenia.
Okay, yeah.
I'm not
sure. So I guess the idea is that people used to sort of think, let's find the gene for, schizophrenia, and let's target this and see what the most sort of explainable mutation is for this, and like do it this way. And then with large scale genetic studies, it kind of has become clear that you need to consider all of the genes together, and sort of there's a confluence of factors which leads to, so if you have like a bunch of different genetic changes, that might lead to the actual sort of development of this disorder essentially in an animal. And so this seems to lend well to like testing those kind of things essentially. There's a big problem, obviously, like you said, with saying EI balance is the cause of what you see in the behavior of schizophrenia. But you can definitely use this sort of technique to say what leads to EI imbalance in these models. And you can try a bunch of different, you can imagine like having hundreds of little organoids growing all with different sort of genetic profiles and seeing which one leads to EI imbalance and which ones don't.
Yeah, or just kind of, if you have the stem cells from a bunch of people with autism and you grow these organoids, what do the organoids have in common? Like whatever it is, even if it's not related to pre-existing hypotheses, it's kind of a step removed from the genetics because it's so hard to just look at genes and be like, oh yeah, I see what's going on. I think that's something we thought we could do once we like had the full genome. We could just be like, oh yeah, now it makes sense. But kind of just letting the genes do their work and see what they turn into is a much easier way to try to diagnose, what all these people have in common and why they have a disease that seems similar.
So yeah, I mean, this returns to the theme that I was trying to get at earlier, I think. And it's an interesting one, right? In the sense that, at one point in time, we might have hoped, we'll be able to grow humans in a dish, or we'll be able to clone humans even without, placing them in a uterus. And the refined version of that we're getting at is, we can take specific subsets of cells from a human and grow parts of humans that don't function like humans in a dish. And similarly, another kind of lofty ambition might have been, we could, from the genome, predict how these things are going to grow. And it's that's actually a challenging problem. It's, we were never going to model that in biophysically realistic detail, like how the cell actually grows. But instead, we can use as a model that essentially predicts how genes will affect the development. We can basically use these sort of crude developmental trajectories that we can reproduce in a dish as proxies for that. So in some sense, when we set for ourselves very ambitious goals, sometimes the sort of failures to achieve those goals is manifest through other things that are kind of hedges that we wouldn't have expected. I don't think 30 years ago, when people were thinking either about how are we going to understand how genes work, or how are we going to be able to build in an artificial setting, how will we be able to grow humans or animals, they would have thought, well, we won't be able to do either of those things, but we'll instead be able to grow kind of parts of animals that inform us significantly about those processes. So it feels like an interesting kind of not quite landing the nail on the head, but getting hedging or approximating what we were aiming for.
You shoot for the moon and you land amongst the stars.
Yeah.
Anything else to be said about their use for studying diseases, or maybe more broadly? I guess as a computational neuroscientist and thinking about the types of things that systems neuroscientists usually think about, the questions seem largely non-overlapping, I guess. The actual activity of the neurons isn't talked about much, with the exception of the fact that they could get broadly oscillatory behavior that kind of matches prenatal development. But yeah, for the most part, we're not talking about information processing in these systems.
Yeah, so there was one line of research that I found somewhat interesting and reminded me of, there was a line of research, I don't know if it was 10 or 20 years ago, but animats, I think is what they were called. What people are proposing to do with these kinds of organoids is embed them in control systems. So you can imagine build a remote control car, or build a circuit which produces light, or something like that, electrically powers an LED or something like this, and have the artificial brain, this organoid, control that. This also felt like kind of an interesting, almost salvaging of a failure case. It's if it turns out that we can't figure out how brains work, and we can't build neural networks that can control some simple control system, which I think we will be able to, so this is not not necessarily a failure case, but we could just grow brains that solve the problems that we want, right? You want to have a control system for a small helicopter to make it an autonomous helicopter or drone or whatever. You could maybe grow a small organoid brain that you train via biological reinforcement learning or something like this to control this helicopter. So it's a way of almost using biological computation. I mean, it at least aspires to this. I think right now it's more almost an artistic novelty, rather than something that would work functionally for engineering purposes. but I'm at least intrigued by the idea that people are trying this, and I'm kind of, maybe part of why I bring this up as well is, again, and I don't want to sound too negative. I think this is an interesting general area of research, but like as almost kooky as it sounds to say, let's not use a computer, let's plug in an organoid to control this control system. That almost
just have a kid instead, and then just train the kid.
Well, I mean, I think we're thinking like, I don't know, make a satellite, but instead of having a computer control, the satellite's probably a bad example because you would still need it to be like in an oxygen and nutrient dense. It's the really bad example.
They did send these organoids into space to see how they would grow there.
Yeah. But that was not to use them as control systems.
As far as we know.
Yeah. So I mean, okay, so the sending them to space is like almost a pessimal example because it would be a completely terrible use case. But one can at least speculate on, wouldn't it be fun to have a computer, an organoid functioning as like a microcontroller or a small computer rather than using electronic circuitry? I mean, because that is what brains do. Brains control things. And I find the kookiness of that in some ways almost as kooky as the premise that these are going to be perfect models of animals in other respects, like for disease processes.
So first, I guess we should say that, so in the New York Times article, which was mostly profiling the work of a lab at UCSD, Alison Muotri's lab, they do talk about a situation where they basically hooked up an organoid to kind of get electrical impulses from mechanical sensors, and also its own electrical impulses could control the output of that thing. So they were kind of trying to embody the organoid. And that's something that I was thinking about when reading this, is like, it's never going to learn to do information processing because it doesn't get any information to process and has no reason to process any information. So I think that is a relevant point to be made about kind of the limitations of these things if they're never going to be connected to a body.
Also the proposed experiment at the end of the first review, which was essentially going further and saying, like, let's use optogenetics to give some photoreceptive cells in there as well, or like neurons that are sensitive to light. Basically, you can very easily, well, not very easily, you can very finely control the sort of stimulation that you're giving to the neurons that are in this organoid, and you can. You can't do information processing, as in it's not receiving input from an external world, but you can definitely look at how neural circuits in that organoid will respond to certain types of patterned information.
Yeah, if you're given it enough, if you give it enough, patterns with specific structure, then that's information. So that's another way to do it too, is to just kind of artificially stimulate, even without interacting with the real world.
I think it's a lot better than our simulations in a computer, right?
Well, that's a question.
It is a question, and I think the answer is yes.
Well, so the same way I would say right now, it's kind of unambiguously clear that artificial neural networks can do more for control than these systems can.
Yeah, but if we're talking about sort of basic neuroscience research, not basic, neuroscience research which is trying to be clinically relevant, basically. So the kind of simulations where you...
Okay, so for clinically relevant things, I would agree. Yeah, if we're concerned more about how cells grow and die, then obviously organoids are more relevant than most of what's been done in terms of computational modeling.
Yeah.
If we're talking about how neurons function for control and for information processing, it's not clear to me that, I would say it's pretty clear that these are nowhere near as useful as artificial models, because artificial models can only
at the moment they're
but so the potentially the optimism, right, would be that you can start plugging these into like bodies, but then and this again goes back to what I'm getting at. It's like, then they're just animals. Like, if you provide a body for an organoid and you have the organoid grow according to its own rules, then it inhabits a body, becomes embodied with a body.
But you don't need to provide a body to do the sort of experiments that gain a lot of information about how the neural circuits process, certain stimuli, right?
Well, you may need to have natural outputs for it. So, like, it's one thing to say, we're going to start showing videos to our cortical organoid. Mm-hmm. But if there's no thing that the cortical organoid has to do, it may not, learn anything.
Yeah.
And when it's just shown videos. I guess you might say, I think that the cortical organoid circuit that exists is going to be capable of some sort of unsupervised learning. and maybe it will learn just when I show it videos. That would be remarkable, right? If that happened, then I would immediately say, oh, this is like a remarkable model. If somehow it could memorize patterns that were shown to it, even divorced from a task. Because this would also start implying that like there are innate circuits that are responsible for, learning and memorizing just in an unsupervised way.
Yeah.
Possibly in Cortex, which would be sort of validation of simple theories of how Cortex might be responsible for actually learning.
Okay, so this is so far you're not surprised if this organoid.
Well, but it's useful. So surprise and cool and useful, these are all different things,
Sure,
right? Like
yeah.
it's kind of cool. I accept that.
Yeah, but
I'm not
surprised,
necessarily surprised.
Yeah.
If it was able to produce something that we are unsure scientifically, what is innate or not in animals. Like if, for example, it could do unsupervised learning, I would say, oh, that's really cool because this actually tells us something about how normal brains probably function. We weren't sure that unsupervised learning was done in a circuit like this.
But I think, have they not done experiments in cultures where they sort of stimulate, where they do some sort of vague associative learning?
Yeah, obviously, like the rules of kind of normal Hebbian learning are studied in culture a lot. So you do get this thing where, like, if you stimulate neurons in certain patterns, they'll start to form certain associations or whatever. So that can kind of happen. But yeah, I can still see the idea. Like, in terms of development, we don't know when things become possible and all of that. So it could still be interesting. I feel like their comparison to artificial mathematical models, I think of as a different type of comparison. It's not about what can each of them do right now, like AI versus an organoid. Yeah. That would be a cool battle. But more just like they are almost analogous in the sense that they are simplified systems that we have way more control over and way more access to than the full brain. So that's kind of the interesting bit to me. It's like if you were going to build... Like the closest thing to a mathematical model in experimental neuroscience is like a culture where you get to control it and record from it and all of that.
Yeah, so again, I'm agreeing with a lot of this, but like one thing that when we think about utility, so again, utility for diseases is different than like utility for control.
Sure.
If we could figure out a way to grow, this is kind of what I was getting at. Like if we could, one of the things I was getting at, if we could figure out a way to grow organoids such that they could be used to like in lieu of a computer, that would be kind of useful. But your response was like, why not just have a kid and train it to do something? And like that's kind of the right answer.
Yeah.
Like
you're basically making like a hybrid kid.
You're making like a cyborg or a cybernetic organism.
I really feel like this is like we're supposed to be talking about ethics now. We keep like skirting around the issues that came up in like the ethics conversations.
Yeah, clearly the technology is not there. But if it were the case that these organoids could control things, then you would really start questioning like how different are they than animals? And they're sort of so obviously not there that we don't need to be having this as an ethical conversation now, though we might be anticipating, we're not the only ones anticipating this. Of course, this is
if you're listening to this in 30 years, we were wrong.
The organoids have taken over.
But the counterpoint would be, of course, that like artificial systems that we can sort of read and write with to computers are easier for us to reproduce and control.
Yeah.
So like the disadvantages of using an organoid as a control system are the disadvantages of using like an animal as a control
There is a
system.
startup though in California that is trying to use, I don't think it's organoids, I think it's just cell cultures as olfaction detectors for different perfume tests maybe or some sort of
There, I guess the claim is that like somehow
we haven't solved olfaction yet.
The sensors are very good. The quality of the sensors are very good.
Yeah.
Okay, right. So like the standard ethical issue with these, one is like, oh no, it's human cells and it's human brain cells. Is it like morally relevant and stuff? And I have to say, I was very impressed. So both the New York Times article quotes a bioethicist named Janine Lunshoff, I think, and then this other interview that we read with the bioethicist Sarah Chan, and both of these bioethicists were like, guys, chill. Like, it's not a big deal. These aren't anywhere close to human. They're not anywhere close to any situation where we should be concerned about consciousness. They're like, it's fine. Like, just keep going. And that's even like in contrast to a a scientist quoted in the New York Times article that was like, what if we put in cells that sense pain, or what if they form memories? It's like, one, they probably are forming memories because they have synapses that can change, and two, what do you mean like a cell that senses pain? It's not like a single cell that like now means you have a conscious experience of pain. So I was just like very happy with the fact that the people who are actually supposed to be the professional ethicists like saw this for what it was, kind of straight out, like it's okay.
Yeah, I mean, there are bigger issues in neuroscience research regarding ethics that, I mean, you know, primate research, any type of animal research, I presumably has much more weight behind it than the question of organoids at the moment. Anyway.
Yeah, so just to play devil's advocate, because of course I agree with what you're saying. I mean,
I was very surprised.
No. I'm just thinking, because I'm being sort of slightly pushing things a little bit further. You know, one of the arguments sort of that happens within maybe, let's say, the vegan community and comes up occasionally is like, well, how do we count bivalves? Do clams or oysters count as things that are possibly conscious?
That's fine, but clearly in the context of neuroscience research, we use animals like primates.
Yeah, so this is a weaker one, but I want to explore the possibility. I mean. So certainly when philosophers talk about it, you know, you might say, like, in order to, if you wish to be a vegan, if you wish to, you know, not consume conscious beings or harm conscious beings at all, you should avoid oysters because there's a chance they're conscious. But, like, realistically, that chance is very, very slim. So it depends on what your sort of risk profile is in terms of damaging consciousness. And I'm going to throw it out here that if these are much larger groups of neurons, perhaps in some cases, I don't know the exact cell counts, but I mean,
I think hundreds of thousands.
Yeah, so this is bigger. This is potentially more neurons than in an oyster. I mean, certainly they're like at least.
It's like a fruit fly, I guess. Hundreds of thousands, right?
Yeah, I think that's about right. It's obviously not about count. It's not just about count. It's about whether they are sort of in a certain kind of structure. But like, because oysters are these largely sedentary, I mean, they don't really move, and they do very little. I mean, this is why you might consider them borderline a vegetable in the first place. I mean, they're obviously animals from a taxonomic standpoint.
But then, yeah, that would kind of hold for the organoids. They don't move. They don't do much.
Yeah. So I'm bringing this up as a sort of purely as a thought experiment. I mean, I do eat oysters, so I'm
you would also eat organoids then?
Well,
yeah.
I mean, are you a cannibal if you eat?
What if it's your own stem cells? Is it cannibalism if it's yourself?
But you clearly have no problem eating like a piece of your cheek or something if you accidentally bite it.
Not intentionally.
No, not intentionally, but I think it's a little bit complicated. I mean, it's certainly right now when it doesn't serve as a control system, or if it turns out that a cortical organoid could serve as a simple control system. Like maybe it could like do something complexity equivalent to like regulating a thermostat or something like this.
Then you wouldn't eat it.
No, well then I would say, oh, it's demonstrating the level of behavioral competence, maybe roughly similar to an oyster. And then I would put it at like maybe we should think about it on par ethically.
So we shouldn't eat our thermostat.
Ooh.
Because we get cold.
Well, I mean, I think that a simple biological control system that could perform equivalently to a thermostat wouldn't be conscious, I suspect. And we wouldn't necessarily need to give it so much moral worth.
I get the point. Broadly, where are we placing things? How do we decide? I mean, these are the questions of bioethics. But the bioethicists have spoken, and we're just not there yet with organoids. That's what they decided. And I like that there wasn't this, I think for like more of a lay audience, there's this sense of like the fact that it's human cells makes it all different. And I like the, kind of the acknowledgement that doesn't necessarily have to be the case.
I can imagine that in previous eras in which bioethicists have made certain statements, it may have caused the public to be unduly alarmed. And so I wonder if part of the way that bioethics is responding to this fairly benign situation without much calling of an alarm is recognizing the sensitivity of issues related to human stem cells in the sort of American, certainly at least historical public consciousness. And so perhaps by not drawing attention to it, they're expecting it to not be overregulated in a way that would be damaging to the scientific progress.
Yeah, that's a good point. Yeah, because it's a risk-benefit analysis. You can't just focus on the risks.
Yeah, but can you have like a bank of like thousands or tens of thousands of them? That's, I think, the computational neuroscientist, high throughput experiment. Yeah, then you have to think about like, is 1 million organoids equivalent to
like a full
cow,
human brain, a
yeah.
cow, something. Yeah. I don't
Anyway,
know.
we'll see.
Okay, I think that's all there is to say about organoids.
Well, I think there's a lot more to say, but
we're not going to say. Till next time.
See ya.
Hey, if you're still listening to this, you must really like us. So how about you go to iTunes or Stitcher and rate the podcast? Give us some feedback. You can also go to our website, unsupervisedthinkingpodcast.blogspot.com. You can comment on different episodes, or you could give us ideas for new topics you want to hear about. We would love to hear from you. Thanks.